Step 2, look at the evidence. Now I'm going to have to move rather quickly, but I want to give you one line of evidence that I think is powerfully suggestive of design in biology, and I'll do it in a little detail. I want to consider the phenomenon of minimal complexity in biology. Minimal complexity has now a thriving future, and it asks the question, how simple can an organism be and still exist? What do you need to be alive at all if you're a cell? And this is the theater in which our little drama will take place. Well, the main approach is bottom-up, because that's, after all, what happened in the history of life according to evolution. Non-living things came alive. And we can also come at the problem top-down, by asking how much can we take away and still have cells that are alive in culture? You can think of this like two tunneling crews going through a mountain in Switzerland. They have to intersect if that's going to be a functional tunnel. Well, analogously, these two approaches, top-down and bottom-up, must intersect at the origin of life if the origin of life actually happened by natural processes. Now, the leading hypothesis right now for the natural origin of life is called the RNA world hypothesis, where the first replicating entity was not a whole cell, but rather an RNA molecule capable of making copies of itself. And complexity would gradually creep around that self-replicating molecule, bottom up, heading towards the living state. Now, even though the RNA world hypothesis is this leading candidate, lots of scientists have questions about it. Stuart Kaufman, for instance, whom both Bill and Sahotra know, in a book about 10 years ago said, I doubt this, and the reason I doubt it is the one most rarely talked about. All living things seem to have a minimal complexity below which it is impossible to go. Because there's a lower threshold, if you pass through it, it's over. And he goes on, he says, your antennae should quiver a bit here. All free living cells have at least the minimum molecular diversity of Pleuramona, and I'll explain what that term means in just a moment. That's Pleuramona. Until last year, the simplest free living thing that we knew. Only 500,000 base pairs of DNA, slightly more than 500,000 base pairs of DNA. A very stripped down cell. The culture media requirements for mycoplasma are very rich because it can't, for instance, make any amino acids for itself. But it can serve as a kind of model of a simple cell. Well, Kaufman asks, why is there this minimal complexity? Well, we can do that analysis. In fact, it's been done using mycoplasma. Mycoplasma was one of the first cells, if not the first, to have all of its genome sequenced, all of its DNA sequenced. And this was published in Science in 1995. Now, I realize you can't read to this, but that's the parts list for mycoplasma. Individual proteins organized by their functional groups. And these functional groups, translation, transcription, and so forth, are jointly constitutive of a living state for mycoplasma. Here are some of these individual actors. This is a protein involved in cell division. This is a protein involved in helping proteins fold as they come out of the ribosome. This is a protein involved in the process of translation itself. Now, you'll notice that these are different shapes, different little molecular actors, because in fact the jobs that they're doing are different. And if you went through that whole parts list and go to the protein data bank where I retrieved these diagrams, you'd see that the shapes are different. So we're dealing with a great deal of molecular complexity, even in the simplest free-living thing that we know. Now, you see a parts list like this, and the question naturally arises, how much of this can we jettison and still have the cell alive in culture? And that experiment was done. In fact, it was done twice. Published first in 1999 by Clyde Hutchinson and colleagues, and then in January, they redid it, or they did it over a period of years and republished the results in PNAS in 2006. They redid the experiment and tightened up some of the protocols to account for some open questions. Here's what they did. They took a piece of DNA called the transposon. Now, these are not to scale. This little cartoon is greatly enlarged so you can read the name. Transposons are bits of DNA that integrate themselves randomly into other DNA. Well, what happens in this experiment is if the transposon lands on an essential gene, it will disrupt its reading frame and you'll get a signal. If that's an essential gene, the cell will die. Well, if you do that often enough, in large enough populations, you can get a pretty good map of which are the essential genes. Because the mere fact that you find a transposon here in a cell that's alive in culture tells you that that transposon did not land in an essential gene because there the cells are still alive, whereas these have no progeny. Starting with these populations and working your way backwards, you can get a pretty good map of what the essential hardware is for that cell. Here's what they found. These are now the figures from the second time they did the experiment. 382 of the 482 protein-coding genes in mycoplasma are essential under laboratory growth conditions, including 110 genes of unknown function. Now, when I read that, the figure was lower in 99, it was about 100. It jumped off the page at me. How could there be nearly a third of the set of essential genes for the simplest free living thing that we know, and they're of unknown function? Well, their first conclusion was, we may not really understand fundamental biochemistry. I want to press on, though, to a more interesting problem. A more interesting problem, that is, for evolution. That's a very high percentage of essential hardware that's unknown. And the reason it's unknown is these genes are found only in mycoplasma and in no other microbial group. This has come to be known as the puzzle of orphan genes. Typically, when a single-celled organism has all of its DNA sequenced, between 20 to 30% of its genome is unique just to that cell, or just to that genus, and to no other microbial group. In mycoplasma, those unique genes are essential, and we know this by experiment. The problem this creates for evolution is the size of the genome of the original ancestor of life begins to grow well beyond what would be reasonable, because you have to put so many unique genetic codes in that organism. There is now, in March 2006, no theory that explains how proteins can change, genes and proteins can change so rapidly that any historical signature is lost. Think about this in terms of an analogy to language. If I gave you the job of finding all the words there are in English, you'd get a curve that would rise like this and flatten out. You'd say, Paul, I found all the words there are in English. There aren't any more to find. Yes, if you come in from hip-hop and other sources, but that's it. This curve is not flattened. In fact, this was published in 2003. A more recent study pointed out that despite the large number of bacterial genomes sequenced, this trend of unique genetic words shows no signs of leveling off. In Plasmodium, this is eukaryote now, this is the parasite that causes malaria, 60% of its genome is orphans. I'll get to the rest of the story in my rebuttal period. Thank you.